All right, so I guess on the top of my list, that Jason Flynn is going to be the first speaker here, but I would say I haven't been able to sit in on any of these others. I've been on the National Experimental Biology meeting, but I'm happy to be here today to hear what's been going on campus. I'm a faculty in the College of Medicine, Microbiology, Immunology, and Molecular Genetics, and also Associate VPR in the Central Research Office. So happy to be here and look forward to hearing what you've been working on. So Jason, are you ready to get going with your screen?
Yeah, I'm ready. I think Jesse might have like some baseline things to talk about first though.
I just needed to remind everyone.
Oh, okay, sorry.
Oh, you're good. I'm so glad you're excited, Martha. So this is just a reminder, this presentation is being recorded. All of these videos will be uploaded to the YouTube channel, and we will be sharing these links. Questions are encouraged from both our panelists and our attendees. The attendees will simply type in your questions for our presenters immediately following their presentations using the Q&A feature in your toolbar. For our panelists, you all will simply raise your hand and unmute yourself to ask your questions, okay? And Dr. Peterson will call on you all. All right, now without further ado.
So Jason, why don't you go ahead and get your stuff started? Looks fun.
Will do. So hello everyone, my name is Jason Flynn. I'm an undergraduate in the Schneider Lab, where we study mechanosensation, which is the sense of touch, and the development of touch sensors in ducks. So ducks are actually a great model organism to investigate, as their bills actually closely resemble the human fingertip and how touch works in that. So during my time in the Schneider Lab, my focus has actually been on projects to analyze morphological and structural differences between various duck species with different foraging habits. My goal this semester was to quantify structures in the bill skin called corpuscles that can detect touch. So how do animals like humans and ducks alike both detect touch? So on the left, you can actually see this diagram of haired skin, and in the skin, there are sensory neurons that can detect touch. Some of these sensory neurons are just free nerve endings, while others are encased non-neuronal structures called corpuscles. So these corpuscles can actually help animals understand the more delicate and defined features of touch that we rely on. So in the actual bill skin, which is glabrous or hairless, these corpuscles can detect touch, and that's based on different pressures and vibrations the duck might encounter. So once it encounters these different touches, it will send the signal towards the brain through the trigeminal ganglia, and the PRV, which is the region of the brain that primarily receives these touch signals, and sends it to the forebrain for interpretation. So my project actually focuses on these corpuscle structures in the skin, as you can see here. So, what do these structures actually look like on the cellular level? And in this paper by Berkut in 1980, he actually imaged these corpuscles that we care about. These are the two of the main type of touch sensors in the skin. One type is called the Hertz corpuscle, another type is called the Granger corpuscle. In mammals, there are also homologous corpuscle structures that resemble these two, called Pacinian and Meissner, but since we're focused on ducks, I'll use the avian terminology. These two corpuscles are very different when you're looking at them. The Hertz corpuscles are much larger than the Granger corpuscles, and these Hertz corpuscles contain lamellar cells that surround the center sensory neuron that's encased in this corpuscle. Whereas this Granger corpuscle might have multiple endings to the same neuron that are encased in the corpuscle, with Granger cells that can sandwich these endings within it. So if mammals also have these structures, as I mentioned, why are we using ducks instead of the common lab organism like mice? So as I mentioned earlier, the duckbill actually closely resembles the human fingertip. The duckbill has both the Granger and Hertz corpuscles in it, just like the human fingertip has both of the mammal homologs in it as well. Whereas if you looked at the glabrous or hairless skin in mice, such as their paws, they only have one type of these corpuscles in a very low quantity. So the duckbill is a great tool for studying these different touch sensors. So these ducks also resemble different foraging strategies. Some ducks might heavily rely on touch to where they dabble, and they can kind of flutter their bill in the water to actually filter feed through different particles that might be there. Other ducks might be divers, where they might filter feed underwater, and they might use visual cues to catch and chase down food. So how might the structures within these bills differ based on the different foraging strategies the ducks might have? So to look at that, I analyzed these micro CT scans of duck bills from various species of ducks, and these are embryonic bill skins, or bills. So in these bills, there are these sensory pits that contain these extensions of neurons that can either be encased in herps or Grandry corpuscles in the dermis, or they might extend all the way into the epidermis as free nerve endings. So our thought here is that the more sensory pits these bones might have, the more sensory touch-sensitive neurons might exist in that species, which might be related to its foraging strategy. So I looked at 3 bills of different dabbling species and 4 different diving species, and we quantified the number of sensory pits in those organisms. While the dabblers had a higher number of sensory pits, and the divers overall had a lower number of sensory pits, we only had a small sample size, and these numbers aren't actually too different, especially since other data we already know, such as the ruddy duck having a high number of touch-sensitive neurons. This data is kind of inconclusive and kind of shows the confusing nature of what actually will show if an organism is touch-sensitive or not. So to actually explore this question further, we went down to the cellular level and tried to actually quantify the corpuscles in the bill skin. So going back to that Berkut paper from 1980, he noticed that these 2 corpuscles, herps and Grandry, were actually at different levels within the bill skin. So here at the dorsal bill, herps corpuscles might be in higher number towards the tip of the bill and lower number away from the bill. So depending on what tissue sample you look at from the bill, these corpuscles might differ in number. So when we take skin samples, we remove the skin from the bill, seeing this embryonic bill here, and we take these punches of a known size in order to get our skin sample. Once we have that, there are corpuscles in the skin sample. We actually want to be able to label those so we can count them. So how we do that is we have this primary antibody that targets this molecule called TUB3, which is a beta tubulin subunit that is neuronal specific. So it's only in these neurons. If you recall from any cell biology classes some of the audience might have taken, the subunits of microtubules have both an alpha and a beta subunit that composes this protofilament of microtubules. We applied this antibody to embryonic trigeminal ganglia, and we saw it clearly label the cell bodies of neurons and the axons of those neurons. So now that we applied it to neuronal tissue, we actually want to apply it to skin tissue where these corpuscles are present. So we stained these embryonic bill skin samples with that antibody. We could clearly see the herpst and the Grandry corpuscles by both using immunohistochemistry, which stained that protein brown, and the immunofluorescence where the protein would fluoresce instead. And we can clearly see the large herpst corpuscle here and the Grandry corpuscle here with the multiple endings inside. So what does this actually look like in adult skin? So we also did immunohistochemistry with the same staining technique in adult skin. And we could see that the herpst and the Grandry corpuscles were still labeled, especially their sensory neurons inside these corpuscles. However, one key difference between the two is that the herpst corpuscle especially, the lamellar cells do not actually express that TUB3 protein as much when it's in the adult stage of life, therefore showing that the TUB3 is actually transiently expressed between these 2 stages of life in some quantity. So my project is actually focused on quantifying these corpuscles. So we would take those skin punches and actually section them, keep only so many sections, and actually count herps and Grangee corpuscles in those sections. And by counting just a few of those sections, we can assume that the entire punch is the same throughout of it. And we can assume that we can calculate, based on that known area, how many herps and Grangee corpuscles are in each of those. So my project is focused on comparing bill skin samples of these different species based on their foraging strategies, but I haven't been able to focus on that just yet because we're actually applying this counting in another project. So currently we're quantifying corpuscles in adult bill skin to supplement a transcriptome project that our master's student Thomas is currently working on. The lab is curious about the actual mechanosensitive channels in these corpuscles, and by quantifying corpuscles in this adult samples, we're actually going to be able to supplement what he finds from his RNA-seq. And this method can also just be used generally to see differences between embryonic and adult tissue. So I just want to thank the Schneider Lab, Dr. Schneider, Thomas, and also want to thank the biology department for all the help over the years. So thank you, and let me know if you guys have any questions.
Thank you very much. That was exactly 10 minutes. Good job. You must have practiced. Are there any questions from any of the participants? Let's see.
Jason, this is Jessie.
Jessie.
I have a question.
Yeah.
It was going to take me too long to type it. So while you're quantifying between the two different types, what has been the most challenging aspect of this process?
Honestly, it was just kind of getting the protocol down for actually staining with immunohistochemistry. So in those pictures where we were staining the protein with that brown color, it was actually trying to get that protocol down to actually stain correctly. So we had this kit that we had from this one supplier, and we had to kind of use our own reagents to kind of guess our way through and actually get a protocol that was working. Now that we have that workflow down, it's been working really well.
There's actually a question in the Q&A from Dr. Jaromchik. Said it's very interesting. He feels much more educated to watch the ducks now in Jacobson Park. But his question is as to whether you use any computer tools in your data analysis.
Yeah, so actually during that bone morphology part of the presentation where I was quantifying those sensory pits, I actually learned how to use ImageJ, or the free version called Fiji, to actually use those stacks of images to actually quantify those micro CT scans. And I think we're also going to be using the same program to quantify potentially free nerve endings in some of these samples to supplement that transcriptome project. So ImageJ is the main tool we've been using.
All right, great, thank you. We probably need to move on, keep on time. And the next presenter is going to be Zoe Hert and Carly Karrick. I'm not quite sure how you are doing that organizing, but I'm sure you've got it figured out. So whenever you can get ready to share your screen.
I'm good to share.
All right, take it away.
Okay, hello everyone. Today we'll be presenting about our study into the variation in the factors driving gut microeukaryotic and prokaryotic diversity in wild lemurs, and my name is Carly Karrick.
And I'm Zoe Hert. So to begin this presentation, we wanted to provide you all with a brief introduction to the gut microbiome. So the gut microbiome is defined as the diverse community of prokaryotes and microeukaryotes within an animal's gastrointestinal tract. And so much of the previous research into the gut microbiome has predominantly focused on these prokaryotic members. And in fact, in wild lemurs, which is our study system, gut prokaryotic diversity has been found to be primarily driven by host diet and host evolutionary history. However, the gut microeukaryotic community is comparatively understudied, so the relative importance of these host ecological and host evolutionary factors remain unclear. And so in order to further assess the factors driving gut microeukaryotic diversity patterning, lemurs provide an ideal study system due to their unparalleled ecological and evolutionary diversity, which is a product of their adaptive radiation on the island of Madagascar.
So we had three primary goals with our research. The first was just to compare the host evolutionary and ecological factors that drive the diversity patterning in the microeukaryotic and prokaryotic communities in the gut. And we also generally just wanted to advance our understanding of the gut microbiome and its diversity. And lastly, we wanted to describe which microeukaryotes are present in the lemur gut. And so to accomplish these goals, we utilized fecal samples from 11 species and 3 families of wild lemurs. And you can see in figure 3 a map of the 4 different sites where fecal samples were collected in Madagascar. And for the microeukaryotes, we sequenced the V9 region of the 18S rRNA gene using PCR. And in order to compare this to the prokaryotic members, we utilized an existing data set that used the same fecal samples, but sequenced the V4 region of the 16S rRNA gene. And we did all of our bioinformatics analyses in QIIME2.
And so the first of these analyses was a measure of alpha diversity. And alpha diversity is essentially a measure of species richness. So it's measuring how many unique microbial species are present within the gut microbiome. So as you can see in figure 4, we have a comparison of the alpha diversity of prokaryotes in red to that of the microeukaryotes in blue. And so we found that lemur taxonomic identity seems to correlate to and explains the gut prokaryotic alpha diversity patterning, which is supported by a very low p-value. However, lemur taxonomic identity does not seem to explain this gut microeukaryotic alpha diversity patterning.
And so the second analysis we did was beta diversity, which is primarily concerned with the who of the microbes. So it's a composition-based analysis, seeing which different species are present in different samples. And in figure 5, you can see PCLA plots of weighted unifrac beta diversity with the microeukaryotes on the left and the prokaryotes on the right. So looking at the left with the microeukaryotes, you can see that there's really no clear clustering or patterning based on host evolutionary history or the ecological factors that we tested for, so host diet and habitat. And those results are supported by Permanova tests of significance. And this also is a really stark contrast to the figure on the right with the prokaryotes, where you can see this really clear clustering based on host family. So you can see how host evolutionary history seems to shape the beta diversity of the prokaryotic community, but we don't see that same pattern with the microeukaryotes.
And our final analysis we have presented here for you all today is in figure 6, which was our measure of relative abundance of various gut microeukaryotes. So within this figure, you can see on the legend in the left, various gut microeukaryotes listed that we found in relatively high abundance across samples. However, when you're looking at the plot, there seems to be no clear patterning of the gut microeukaryotes according to lemur host. And so from this information, in addition to our literature review investigating these various microeukaryotes, we find that the gut microeukaryotes most likely represent transient members of the gut microbiome. So these microeukaryotes may potentially be a dietary or environmental component, or even a pathogen of a given lemur.
And so taking into account all of these results, we can conclude that neither host ecology nor evolution drive the microeukaryotic diversity in the gut, whereas these factors do seem to shape the gut prokaryotic diversity with an emphasis on host evolutionary history. And like Zoe said, we can kind of use this conclusion to think that maybe these gut microeukaryotes are transient members of the microbiome, so they're just coming and going. And if they're not performing as important functions for the host as the prokaryotes are, then they would be less essential to host health and overall fitness, and therefore wouldn't be retained by things like ecological filtering and natural selection.
As we mentioned at the beginning of this presentation, the gut microeukaryotes are relatively understudied. So we hope that through the popularization of this new area of research, that improvements can be made to various microeukaryotic databases so that we can in turn improve the detection of parasites and other functionally important gut microeukaryotes, so we can better elucidate the gut microeukaryotes' role within the gut microbiome and their influence on host health. And finally, this research is directly applicable to conservation initiatives, as the gut microbiome, particularly through the usage of the DNA metabarcoding method, can rapidly assess organismal and population health. And in fact, this is particularly important for lemurs, as the IUCN considers lemurs to be the most endangered group of mammals. And so by understanding lemur gastrointestinal health and population health, we can develop more effective legislation and practices to conserve lemur biodiversity for the future. Thank you so much for listening to our presentation. And at this point, we'll take any questions.
Thank you very much. That's fascinating. I have a question. Who gets to go to Madagascar to do these collections?
Mariah Donahue primarily. She helped a lot with the, mentoring us and also, collecting all of the samples for us.
Okay, so that's somebody here at UK.
Yes.
So and they said that there are 11 different species, but only like 1 or 2 samples from each species. Is that?
So we have a bunch of different samples for each species. It's just 3 families of the lemurs.
Okay. Okay.
Yeah.
So did you find it surprising that you weren't finding anything consistent with where on the island they were living or diet, or maybe they have all similar diets, or I mean, you were looking for differences but really didn't find them?
Yeah, the lemurs have a huge array of diversity in their diets as well as their environments that they inhabit. And so based on previous microscopy-based studies, we really expected to see some stark differences between at least social groups, but we continue to not find any significant results in that type of patterning.
Curious. So are there other questions? I can't see raised hands. So if somebody has a raised hand, feel free to. Maria Donahue says, amazing job, you two. So
I see a question from Dr. Cooper. So yeah, we did not personally collect the samples. They were sent to us, and in the wild, the fecal samples are normally only on the ground for a few seconds. We're following the lemurs throughout the day. So as soon as they, defecate, the fecal sample is picked up as soon as possible. So that way there's no influence of the soil or anything on what we detect.
Good. Let's see. All right. Anybody else have any questions? Okay. Well, if not, thank you both very much. That was it. I'm glad somebody gets to go to Madagascar. So then the next speaker is Cameron Howell. And yeah, here's his slide. And Cameron will.
I'm ready to present.
Excellent.
So, can everyone see that?
Yes, we can.
Okay. So good afternoon on this rainy day. My name is Cameron Howell, and I'm going to be presenting this poster about the effect of 2-week treatment with a hemp extract, NCMB1, on lung tissue in Caribbean vervets with induced pulmonary fibrosis. To get started, here's some background information. First off, previous studies in mice have demonstrated that cannabinoids derived from hemp, especially cannabidiol or CBD, have potential as a therapeutic agent for inflammatory lung diseases. COVID-19 is a prime example of one of these inflammatory lung diseases. A recent in vitro study, which means it was conducted with a test tube or a culture dish in human small airways epithelial cells, reported that a hemp extract known as NCMB1 decreased expression of pro-inflammatory genes and increased expression of anti-inflammatory genes relevant to COPD. Chemical analysis of NCMB1 indicates that the extract is a mixture of different cannabinoids, including CBD, like I mentioned earlier. As a follow-up to this study, an in vivo study, which means it was done with living organisms, was completed by our lab in the summer of 2018 to determine the effect of NCMB1 on lung function in Caribbean vervets with previously induced pulmonary fibrosis or scarring of the lungs. The lungs of these vervets exhibit significant homology with those in humans, so it makes them a great translational model for human disease. In this study, 16 male vervets were evenly distributed into 4 groups, and each group received one of the following treatments in banana slices, twice daily for 2 weeks. The treatments were a control, which was just corn oil, undiluted NCMB1, a 5 times dilution, and a 25 times dilution. Tidal volume, inspiration flow rate, and respiration rate were measured before treatment, at 1 week, and at 2 weeks, and these results are shown here. In figure 1, we have the results for tidal volume, which is the volume of air that moves in or out of the lungs with the normal breath. So you can see with the 5 times diluted group, there was a significant increase in tidal volume after 2 weeks. In figure 2, we have the results for inspiration flow rate, which is how fast air enters the lungs. So for both the 5 times diluted and the undiluted groups, there was a significant increase in inspiration flow rate 2 weeks after treatment. Finally, in figure 3, we have the results for respiration rate, which is how often breathing occurs. So for both the 5 times diluted and the undiluted groups, a significant increase in respiration rate occurred at both 1 week and 2 weeks after treatment. So overall, after 2-week treatment with 5 times diluted NC-MB1, lung inspiratory function increased and airway resistance decreased significantly without adverse effects, while the results following treatment with the undiluted NC-MB1 were less clear. Also, treatment with 25 times diluted NC-MB1 seemed to not affect lung function at all. So the goal of my project specifically was to perform a histological assessment of the lung tissue from these vervets in this study. Cannabinoids typically function by binding to cannabinoid type 1 and or type 2 receptors. So we hypothesized that treatment with NC-MB1 may enhance expression of CB1 and or CB2 in their lung tissue. So following this study I just described, complete necropsies were performed, which is just another term for autopsy, and different tissues were stored, which included lung tissue. So lung tissue from control and treatment groups was dissected, stained, and blindly evaluated for tissue integrity and or damage. Also, immunohistochemistry was performed to indicate the presence of CB1 and or CB2 receptors within various lung conducting segments. These results are still in the works, so I don't have anything to show yet, but we predict that CB1 and or CB2 receptor stimulation may have increased their lung function significantly without altering the histological integrity of lung tissue. There are many directions for this research to go because this was a pilot study. First off, we need to determine expression of genes related to inflammation in the lung tissue, which would complement the in vitro study I mentioned earlier. Second, we need to perform histological assessment of other stored tissues, especially heart tissue, due to the close nature between the cardiovascular and the respiratory systems. Third, we need to identify the active ingredients in NC-MB1. And finally, we need to replicate the experiment with female vervets, a narrower dosage range, like 3 times diluted to 10 times diluted, and more time points. In conclusion, I'll just like to take a little time to thank my mentors, Dr. Osborne and Shay Sickles, for their mentorship. Does anyone have any questions for me?
Sorry, have to unmute here. That was really nice. Let's see. Okay, and no new questions here, but well, let's see. I'm not seeing one. Okay, so this is really interesting. And I guess you were giving this orally, right? And then so this is going to be a systemic thing, and one of the things you're measuring is the lungs, which is great. So it's good that you've got the other tissues to be examining there too. But has there been any consideration of trying to give this as sort of an inhalation method?
I'm not sure because I'm new to the lab. So I started research at the beginning of last month due to the pandemic. And this study was conducted in 2018. So I'm not sure.
I see.
Also in this study, they measured body weight, fasting blood glucose, blood pressure, and heart rate. And there was no significant differences in any of the groups.
Okay.
Except for the 25 times diluted group. At baseline, they were hypertensive. And then at 1 week and at 2 weeks of treatment, there was a significant decrease in their blood pressure. So there's a potential anti-hypertensive like treatment there that that could be explored too.
Yeah, well, these are really important studies since all of this stuff is now legal and nobody knows how best to use it. So
Yeah.
I was the respiration rate. I mean, it seems I could see the tidal volume and the inspiration flow that your lung function is better, but I would seem that you might not want your respiration rate to be increased. I mean, that means like you're breathing faster than is that maybe not what they're measuring?
Let's see, these monkeys, before this study was done, they were induced with a moderate level of pulmonary fibrosis. So I think an increase in respiration rate it could be interpreted as something positive because they've had their lungs damaged.
Yeah. Okay. Are there any other questions?
Dr. Cooper asks, are you measuring these breathing volumes in awake monkeys or are they anesthetized?
That is a really good question. And that is something I'm not sure of, but I think they may be awake when they were measuring this.
All right. Well, thank you very much.
Thank you.
Okay, sorry. There we go. All right, so our next speaker is Vaaragie, if I'm saying that right. I'm sorry if I'm not. Subramaniam, who worked with Robin Cooper.
All right, is it sharing now? Because everyone can see it?
Yes, we can.
Okay, all right. So hello, everyone. So I am presenting my project on bridging optogenetics, metabolism, and animal behavior for student-driven inquiry. And I worked under the mentorship of Dr. Robin Cooper in the Department of Biology. So temperature affects metabolism, and this is commonly known. And this is because metabolic pathways involve the use of enzymes, which are temperature dependent. So the higher the temperature, the faster the enzymes are able to catalyze reactions and thus produce more ATP at the end. Because there's more energy for the organism to use to power all the things that they need to do to survive, the rates of development are also increased when temperatures increase. So both metabolism and rates of development are temperature dependent. Another process that is temperature dependent is optogenetics. So optogenetics is a technique that is used that involves the use of light and genetic engineering to study gene expression in organisms. And so the reason why optogenetics is temperature dependent is because it relies on the system called the Gal4 UAS system. So when you're trying to use optogenetics to study organisms, you have to genetically modify the organism so that they have the Gal4 gene and a UAS sequence, which stands for upstream activating system or sequence, which is just a DNA sequence that the Gal4 protein can bind to. So the Drosophila that we used, or fruit flies that we used in this experiment, had motor neurons that had the Gal4 gene and UAS sequence, which was paired with a reporter gene called channelrhodopsin. And channelrhodopsin is a light-sensitive ion channel that can be activated by blue light. So what happens is the Gal4 protein gene will be expressed within the motor neurons of these Drosophila, and that will bind to the upstream activating sequence, and that will cause the channelrhodopsin to be expressed and inserted into the motor neuron's cell membranes. And so when blue light will activate the channelrhodopsin, this will cause the neuron to depolarize and causes the motor neuron to have an action potential. And this can be seen as a behavioral change within the Drosophila by observation. The reason why the GAL4 UAS system is temperature dependent is because it relies on protein replication and transcription, translation processes. So because of this, the downstream effects of channel rhodopsin are also temperature dependent. And so with higher levels of higher temperatures, you'll have more expression of channel rhodopsin. The goal of this project is to create an educational module for advanced high school and college students so they can understand the topics of optogenetics, metabolism, and rates of development in Drosophila and how it's affected by temperature. So what we were doing was we created protocols for these different topics and how they are related to temperature, and tested these protocols out to see if we get the proper results that we would expect so that it can be implemented into these educational models. So the first protocol that we created was for the development of Drosophila. And we took Drosophila and we raised them in these vials and put them in these water baths that were at different temperatures, at a high temperature, middle room temperature, and like a lower temperature, and measured the developmental rates of how long it takes for the pupa to form and how long it takes for the adult Drosophila to form. So the results show that, as expected, at higher temperatures, you had higher rates of development, which showed that the protocol was effective in terms of giving us the results we wanted. Next, we measured the CO2 production of the Drosophila at different temperatures. So CO2 is a product of metabolism. So by understanding the rates of CO2 production, we can relate that to the rates of metabolism. This is because CO2 is produced during cellular respiration, specifically during the citric acid cycle. So for what we did over here, we put 50 Drosophila into each of these chambers, and we had a CO2 monitor placed at the top of the chambers. And then we placed these chambers into different temperature water baths and measured the CO2 production over a few hours and then graphed them. So it's a little hard to see here, but the higher temperature had more CO2 production as we would have predicted. So based on these results, it showed that the protocols that we made were effective in terms of showing the results that we wanted. And it showed that the increase in temperatures did cause an increase in the development of the Drosophila as well as metabolic rates. The next steps of this project would be to check the channel rhodopsin expression levels. And this can be done by observing the differences in animal behavior within the Drosophila when they're exposed to blue light at different temperatures. And so if there's a difference the difference in animal behavior can be correlated to changes in the expression of channel rhodopsin. And once we do that and that protocol seems to be effective, then we can implement this module into an actual high school classroom and see if this module is effective in helping students learn about optogenetics, metabolism, and the development of Drosophila and how it's affected by temperature. So I would like to thank Dr. Robin Cooper, as well as Tawny and Brett Criswell for helping me with this project. And now I'll open up the floor to any questions.
All right, thank you very much. So I have a question. You said you're going to be measuring animal behavior. So what exactly are you going to be looking for?
So when you have so if there's more channel rhodopsin being expressed, then that will cause more contractions within the motor neuron. And so if there's an extreme amount of contraction, it'll basically almost put the motor neurons in a state of like so much contraction that's a little bit paralyzed, like it's just like extreme activity. And so that's what we would be looking for in terms of the behavioral aspect.
And so you put each of the different samples of flies under blue light, and based on the temperature you expect, you're going to see really obvious differences.
Yes, because these are really sensitive to the types of Drosophila that we're using are really sensitive to the blue light, so more than usual. So it should show a bigger difference in terms of behavior.
Jesse asks, if you were to repeat this project, what would you do differently?
I would say have more, so there's a lot of different, well, obviously we can have more samples so that we can get more quality results. I would also add more, there's like different things that we can add to the protocol as well, like looking at cofactors of the channel rhodopsin, kind of making it a little more complicated. So I would also add that, and we're in the process of adding that as well. But that's what I would change.
So Dr. Cooper says, great presentation. I can't wait for you to come to high school and college classes with me to try these out. Hint, hint. 30 kids in a high school class, total chaos. Yeah, especially if you're watching jumping or frozen Drosophila. I'm curious, so you would be, one of the modules would be to measure the CO2 in the classroom. So I guess you would be bringing the various pieces of equipment necessary for them to do that, because I would guess not every classroom has one of those CO2 monitor things, right?
Yeah.
Okay, so you envision like spending a day in a classroom and helping them get through the experiment in a day or two?
Yeah, and we also have, we worked with a high school teacher, and so she made some worksheets and things as well for the students. And so getting them to fill out the worksheets and evaluating if they were understanding the concepts after the experiment is also part of it.
Very good. Robin, Dr. Cooper says, yes, a day with many sections, repeated visits, chaos again. Okay. All right. Well, thank you so much. That sounds like fun.
Thank you.
All right. So our next speaker then is Rachel von Ebers.
Hi.
Hello.
Hello. Let's see here. All right, awesome. Can everyone see that?
Yes, we can.
Okay, perfect. So my name is Rachel von Ebers, and my mentor is Dr. Elizabeth Duncan, and I will be presenting today my research poster, which I completed for Bio 398. So Schmidt-Atea mediterranea is a species of planarian worms. It's a freshwater organism that primarily lives in the Mediterranean, but it contains extraordinary abilities for regeneration. And the organism itself is practically immortal. If you cut off a section of the worm, it can grow an entirely new organism without any difficulties, which is extremely interesting and is primarily the focus for a lot of cancer research, because all this growth obviously needs to be contained, and it has a lot of processes to contain this growth. So I was first introduced to Dr. Duncan's work in my honors cell biology class, where she used a lot of her research as examples for our topics, and she really integrated her research, and that's how I became very interested in it. The focus of her research is SET1 and MLL1 and 2, which are major enzymes that methylate histone 3. And her lab identified a bunch of target genes associated with MLL1 and 2. And they annotated this huge list of hundreds of genes, from which I chose PPM1D and its interaction with P53 and oncogene in tumor genesis. So a little bit about histones. Histones are arginine and lysine rich proteins, which DNA wraps around in order to condense the DNA. And the transcription activity of the DNA can be controlled depending on the tightness, if you will, of the DNA around the histone. So to loosen the DNA for transcription, histones or methyl groups can be added to the histones to accomplish this. And MLL1 is a histone methyl transferase that trimethylates histone 3 at lysine 4. And this is really interesting for our cancer research because the histone variant H3.3 has been found in increased levels in brain cells. And this is interesting because this histone allows the brain to transcribe these genes without dividing, which is very important because, as we know, brain cells do not divide as much or if at all as other cells in the body. So we have found that MLL1 through our literature research has also been linked to neurogenesis, which is very interesting. And then a little bit about the gene that I researched. PPM1D is a protein phosphatase, which encodes, the gene encodes for protein phosphatase that is induced by P53, which is a tumor oncogene. And during the DNA damage response pathway, this gene is activated. It acts as a negative regulator, which suppresses cell stress apoptosis. And in brain cancers, a gain of function mutation in PPM1D inappropriately dephosphorylates P53 and CHEK1, a cell cycle checkpoint kinase, and allows for inappropriate cell proliferation. And while PPM1D is extremely useful in its role in these cell response pathways, a gain of function mutation is obviously very harmful and will cause tumor genesis. And throughout our literature research, we found some very interesting studies that show the connection between PPM1D and this histone variant. And this showed that almost always brainstem gliomas that contain this histone mutation also contain a PPM1D or a P53 mutation. So then our research turned to how we can better serve our Kentucky community. And it was found by the Markey Cancer Center here at the University of Kentucky that there is a increased incident rate of pediatric CNS tumors in Kentucky, more specifically an 8% higher rate, which is very interesting. And currently there is no known cause for this higher incidence rate. So part of our research purpose was to suggest a hypothesis for this incidence rate. And I have found with the Kentucky Geological Survey that there is large deposits of uranium and thorium and radon in Kentucky, more specifically found in black shale, which is a rock that is found a lot in Kentucky. And this rock frequently comes in contact with groundwater. So for future research, it could dive into taking samples from pediatric patients with CNS tumors and examining hair, blood, urine samples for increased rates of uranium. So now I will open the floor to any questions.
All right, thank you very much. So it sounds like this was more of a literature review than getting your hands wet in the lab kind of thing. It's an unfortunate timing, these things, but yeah. You muted.
Yes, due to like COVID-19, it was very hard to get back into labs.
Yep.
So.
Yeah, planaria really pretty unusual, but really cool organisms. Dr. Cooper asks, says, nice presentation. Could some of the incidents maybe be genetic in Eastern Kentucky, perhaps?
Yeah, that is something interesting that we considered also. However, it's very difficult to find any literature research on this because it is such an isolated area in Appalachia. No one is really doing a whole lot of research on this currently.
So I didn't realize that there was an increase in pediatric central nervous system tumors in Kentucky, that there was a higher incidence. But I think from our cancer registry, it's pretty complete. I wonder if you could overlay maps of patient, where the patients are from with the maps that they have in KGS for the various areas of the radon and uranium. Do you know that those data sets are even available?
So I am currently looking at correlating the maps of uranium and radon, which are radioactive elements. And there actually is, if you compare the graphs side by side, there's actually a surprising correlation between the two. So that is something interesting to look at in the future.
You know, I don't know if you're aware that there actually is a really big funded project led up by nursing to try to educate people about getting the radon mitigation systems installed in homes and putting radon detectors out there, because you're right, people don't always realize where they are maybe being exposed. So
right, yeah, you can request a free radon test from that study. So that's very cool. Yeah.
Yep. Are there any other questions here for Rachel? All right, well, thank you very much. Oh, wait a minute, great job, Rachel, from Dr. Duncan. To Rachel's credit,
Thank you, Dr. Duncan.
She helped me reach out to the SEER registry to access the data on mutations in Kentucky glioblastomas. Great. And actually, I chatted with Dr. DeRazio, whose lab is right next to mine. And he said that they're going to be starting to do, or at least offer, exome sequencing to pediatric cancer patients to sort of look for some underlying genetic components to pediatric cancers. So yeah, okay, good.
Yeah, that's very interesting. Thank you.
Thank you. All right, then we're going to switch from the biology section over to a few talks from the engineering area. And our first speaker in this group is Joseph Clark. We say maybe, and now for something completely different.
So hello, I am Joseph Clark. I'm a student here at the University of Kentucky in the College of Engineering. I am a computer engineering major and I do research under Dr. Himanshu Thopalial. And today I'm going to talk about a project I did for predicting future stress events in drivers. So high levels of stress impair task performance and can lead to accidents while driving. A stress detection system can help control stress levels by giving information about when you should begin stress mitigation. But stress prediction improves the stress mitigation by allowing mitigation to begin before the subject enters a stress state. And so a lot of research has been done on stress detection, either using simple correlations or using machine learning. But we were unable to find any research which attempts to predict stress before the subject enters a stress state using machine learning algorithms. And so if this were used in conjunction with like a car entertainment system or a smartphone, then the model could trigger an intervention to decrease the subject stress level and prevent it from going above some level. And so the model that I'm going to present is meant to predict the stress level of a subject up to 1 minute in advance. The model has four main stages. There's data pre-processing, feature extraction, feature expansion, and training and testing. So the training and testing phase uses a leave one subject out testing approach. And so with this approach, you select a subject to use as the test data, and then you use the rest of the subjects for training data. And you do that for each subject one time. So each subject is used for testing once. And so the training data are used to train a random forest classifier, which the test data is then used to evaluate the performance of. The pre-processing phase consists of normalizing the input data into a range from 0 to 1, and then we use a Butterworth filter with varying cutoff frequencies depending on the input signal to clean the data. And then in feature extraction, we extract a variety of statistical and frequency-based features from the different input signals. We get a total of 42 features out of feature extraction. And then in feature expansion, which is a little bit unusual, I think, we take n rows of the extracted feature data, and then we take some statistical measures, which are the mean, median, standard deviation, minimum, maximum, and time weighted average of each of the original features. And so from this, we get a total of 252 features, which we use to train the model. So the data that we used here was some physiological data from 17 subjects as they completed a 20-mile driving route through Boston, which were retrieved from an online database where they were posted by the creator of the data set. The data for each subject includes galvanic skin response, respiration, and electrocardiogram or ECG data taken before, during, and after the subject completed the driving route. Each record is broken into 3 distinct driving situations, which is rest, city, and highway. Each record starts with a rest period and then goes city, highway, city, highway, city, and then rest. And so what the model's actually predicting is we're assuming that rest is correlated with low stress, highway is medium stress, and then city is high stress. And so to evaluate the trained model, we measured the F1 score, the precision, and the recall of the model. The results can be seen in the table here, where it achieved solid scores. They're all around 0.9 and the highest is 1. So that's good accuracy performance. And the table here in particular was done with N from the feature expansion equal to 5, which was the highest performance number that we tried. And then there's another graph below where we varied N and measured the performance of the model, specifically the test data accuracy and the F1 scores for the low stress and high stress categories. And as you can see, aside from some unusually good performance in the low stress F1 score at N equal to 2, it has a positive linear correlation with N. And so generally speaking, the model predicts the stress level of subjects up to 1 minute in advance successfully using GSR respiration and ECG data taken from drivers. And the results indicate that the model performs well and could be expanded to include more driving situations or be used as part of the aforementioned stress prevention system. And so there are a few things that could be improved about this approach. So first and foremost, all the data used to train the model was from the same driving route. And anyone who knows machine learning knows that that can be an issue because the model can make incorrect assumptions about how the input is related to the output, more or less. And so we could probably dramatically improve the performance of the algorithm on different roads by collecting data from other roads and trying to generalize the input data. Additionally, the ground truth we used may not be completely accurate because it assumes that the driver's subjective stress level is determined completely by the type of road that they're on, which obviously could be influenced by other factors like other drivers or weather conditions, that kind of thing. And then the last major improvement that could be made is we could use a different machine learning model as the basis of the model. So something like a deep neural network, specifically a recurrent deep neural network, which can account for time dependency in the input data by itself. So yeah, I guess I'd finish up here by thanking Dr. Himanshu Thapla, who was my mentor in conducting this research.
All right, thank you. So yeah, have you ever driven in Boston?
No, I can't say that I have.
It's not a lot of fun. So there you had all these people that I guess it must have been some sort of a special car that was able to measure some of these physiological parameters that you put into your model. Is that how this was done?
Not exactly. They did augment the car, I'll say. They had like they attached some additional sensors to the steering wheel to measure the galvanic skin response on the subject's hands. They put sensors into the subject's shoes to measure their foot galvanic skin response. And then they put a chest strap attached to the seat belt that could measure their chest compression for the respiration data.
And when you say that you could, the model could predict 1 minute in advance, that would be like if they're moving from highway to city, they would sort of start getting stressed, and then when they were maybe on the highway or in the highway and moving to one of the lower stress, that could be detected as well? I mean, is that what you mean by predicting 1 minute in advance?
Yeah, so we basically grouped all the data into the different categories and then took the last, however much time of each category to take data from and then use for the model to predict it.
Okay, we do have a question from Dr. Jaromchik. How big is your data? If you have big data, what other things beyond precision, recall, F1 would you analyze?
So the data set that we had wasn't actually all that big. I suppose it was like, thousands of measurements, but there were only, I want to say like 5 or 6 transitions between stress levels in each set. And we only had like 17 usable records or something like that. So ideally, we would actually go get more data to generalize the model. And I didn't mention this specifically, but preferably rather than assuming that the driver stress level is correlated with the road type, we could switch over to something like cortisol, which normally has to be measured via like a swab and then sent to a lab for testing. But I've seen research recently that someone at a different college has developed like a wristband that you can put on somebody that can measure their cortisol levels, in real time.
That would be convenient for this sort of a thing that you're looking at. Yeah.
Yes.
Are there any other questions? No. Well, thank you very much for your presentation, Joseph. And our next speaker up will be Tyler Cultice. Puff-based security framework. All right, Tyler, are you ready? Yeah, very good. We can see that. Thank you. But you're muted.
Sorry about that. Hello.
Ah, there he
There we
is.
go.
There he is.
Yeah, I have like 30 different inputs on my computer, so I didn't know which one was my mic. Okay. So hello, my name is Tyler Colteis. I am an undergraduate researcher here at the University of Kentucky, specifically the College of Engineering. I am a computer engineering major, and I too did research with Dr. Himanshu Thopalial on puff-based CAN security. So the controller area network bus, or the CAN bus, is essentially a communication methodology or a protocol that allows for multiple devices, generally within cars and construction equipment, to communicate with one another. And these applications use it for 2 major features. One such feature is the fact that it uses 2 wires, as we can see in figure 1. These 2 wires, as opposed to 1 wire, instead of just sending 1s and 0s as normally a sort of wire would, it uses 2 of them where it measures the difference in voltage between them. And this is important because environmental factors such as electrostatic discharge are actually heavily mitigated using this methodology, which is important in a vehicle because you don't want your vehicle to like veer off the road or something with power steering just because it got shocked or something. So the errors are also corrected with CAN as well using this 2-wire system. And the other major factor is that all of the devices, there's hundreds of them within a car at a given time, all communicate just using 2 wires. So that's what a bus is it's essentially all the devices connect on the exact same line, and to filter it, they use identifiers or IDs. And the recipient will just look out for the ID while it's being transmitted on the communication line. And when it finds that ID, then it'll know, yeah, I should take in that data and process it. So there are two major downsides, however, to using the CAN bus. One such downside is the fact that because it has to be so error resistant, it cannot have a large data frame. So generally speaking, security standards use tens to even thousands of bytes, depending on what you choose. And having an 8-byte limit means that you have to use more than one message to transmit the information if you want to secure it effectively. And that comes into play later in the protocol that we've selected. And then the second problem is that, again, the standard has no built-in encryption or security measures. There is absolutely no possible way to, in the standardized form, to stop somebody from just injecting their own device into this network and posing as one of the other devices. Even though there's ID filtering, because nothing is encrypted and nothing is secured, it is very easy for one to just say, I'm another device, listen to what I say. And we see that with various news articles showing that people are hacking vehicles, even older vehicles. They hack it by putting one single microcontroller onto this CAN bus, and they can manipulate the entire vehicle from startup to steering to acceleration and brakes even. So the two major encryption schemes or security algorithms that we use are one is actually a device, but the first one is elliptic curve Diffie-Hellman, and that is essentially a way to generate keys between two devices without ever sharing any sensitive information. So if you think about keys opening a box, imagine that you and a friend are able to create the exact same key without ever transmitting, or giving to each other any information that's sensitive. So the way that you do this is you send sort of a public key, which is a vague, you can't derive the shared key from it. But if you both use each other's private if you use your own private key and the other person's public key, and you XOR them together and whatnot, you will actually receive the exact same key at the end of the day, meaning you can make encryption schemes without ever sharing the sensitive information unencrypted. The other thing that we use is the PUF, or the physically unclonable function. And this is an actual physical device generally within the processor of the little microcontroller. And this device's purpose is to always output the exact same number or the exact same sequence. And that's important because with attacks nowadays on microcontrollers and stuff, people are able to read into memory. So hard coding, as they would say, or just putting into the memory of the device or the ROM of the device the private key that's sensitive information is not sufficient for security. So having a PUF that the device will ask for that output when it's generating the keys is important. And generally speaking, attackers will struggle. It is infeasible to actually try to derive the output from that PUF for their own use to find the private key. And the PUF can also generate a random number. So then you can create a true random number generator, which random number generators generally are based off of algorithms, pseudo algorithms that basically, if you find trends in the algorithm and you know how it works, you can actually find the next random number that would occur and things like that. And because you can do that, if you want a true random number generator, you want something that's based off of hardware, like physical hardware, such as like the temperature reading up to thousands of decimal places and stuff like that. And the PUF can also give a random number generator, and those are all important continuing on. So the way that this framework works is that it runs in a server node authorization form. And what that means is we add one extra microcontroller to the 2-line bus. and its job is to, when the car starts up or the devices turn on, it will handle authorizing, verifying, and determining the integrity of the other devices. So what will happen is when the car turns on, the server will listen for all the devices, all the other nodes, the hundreds of other microcontrollers, to try to communicate to it. And there'll probably be more than one server depending on hundreds, but we'll use the example here in my figure 6, the picture. The 2 microcontrollers will talk to the server node in the center, and it will send an encrypted piece of information that both the server and the like device itself knows, the node. And what will happen is the server will generate the shared key as well and try to decrypt it. And if it decrypts and comes out to that right number, it knows that the other side has the correct shared key, which generally means they have the right public key. And thus it is authorized or whitelisted. And after a certain amount of time, a timeout period, the server will, or the devices will assume that the server is done taking requests and will accept a packet from the server and information from the server on who is okay to connect to and talk to versus who you shouldn't, because it either was invalid, malicious, or whatever. So it actually creates a sort of whitelist of who to talk to, who to not talk to, and whether or not to raise an alarm to the driver or something like, hey, there's something wrong with your brakes or something. And once that happens, the nodes will actually communicate to one another by creating a shared key between each other, and will always encrypt their information. And to mitigate other attacks that people could do, it also includes something called an Hmac, or a hashed message authorization code, which is essentially it's a very unique way of verifying that the data right after it is correct, which is important because if you encrypt the Hmac and the data put next to each other, if the data is unencrypted in an incorrect way, the Hmac should be able to detect that. We also use a counter system. That's where the TRNG comes into place. So the server and the node both have a counter, and they know whether or not the next packet is valid based off of that counter. Because generally speaking, an attack somebody could do is just take the same message that they just picked up on the line and repeat it over and over and over again, and that would break the encryption problem, right? Because they wouldn't need to encrypt anymore. They know what the packet is. But with that counter, they actually can mitigate that by making every single time the server or the nodes communicate to one another, it's unique. It's a completely different thing. And with encryption, it looks completely different too. It's a completely different sequence every single time it communicates. So the results,
Tyler,
Yep.
you're coming up on 10 minutes now. So
Sorry about that. Yeah.
thank you.
Yep.
So yeah, you can continue, but please try to wrap
Oh
up.
yeah, I'll wrap it up extremely fast here.
Yeah.
So we see that based on existing protocols, there's an extreme amount of packets used when it gets to higher amounts of nodes. Ours only uses linear, so it's actually much smaller, as we can see in figure 9. And future directions we could go with are post-quantum cryptography to prevent the breaking of our algorithm with quantum computers, and conversion to CAN-FD, which is an algorithm that, or a new CAN protocol that has larger packet sizes. That is all. I'd like to thank Dr. Thopleel, and I apologize for almost going over 10 minutes.
Thank you very much. Wow, that's complicated. I didn't understand a whole lot of that, except for the fact that we need people like you to make sure that our cars are working now, because you're absolutely right. They can't work with all these things that are talking to each other. That's why those little alarm lights come on your dashboard when your tire's getting a little low because something happened, right?
Yeah.
Yeah. All right. Are there any questions here for Tyler?
Tyler, this is Jessie.
Yep.
I just have a question for you. What exactly are your career goals with this?
Oh, so The lab that Dr. Himanshu Thoplial runs is a lab about doing hardware-based security. So with this in mind, I'm actually going for a PhD in hardware security, generally over stuff like this, maybe not specifically CAN, but the takeaways from the server node authorization and the limited packet size can be applied to multiple different protocols and multiple different communication paths. So while CAN itself is not in newer vehicles, it's very important to realize that when CAN is being used, encryption is important. And when any protocol is being used for something such as important as somebody driving a car, their safety is on the line. Such encryption is important, and being able to find lightweight and non-performance destroying like methods and means of communication is vital. And that's where I generally took this research.
Great, thank you so much. We'll move on to our next speaker, Chase Ballard and Brendon Bultman. I guess we'll be presenting together. So, you guys are ready?
Hello. I think Chase is sharing.
Okay, there's Brendon and Chase. There we go.
All right, cool. Hello, everybody. I'm Brendon Bultman.
I'm Chase Ballard.
And today we're going to be talking about competitive programming and its roles in improving research skills. And so as a really quick rundown, we're just going to discuss a quick problem related to competitive programming, and then discuss how this problem kind of relates to research, and then the breadth of knowledge that you need to compete, as well as how this knowledge kind of applies to research, and then some benefits of competitive programming. So really quick before we dive in though, what competitive programming is it's essentially a competition in like the software engineering, computer science world, where you're given a set of problems and you have to solve them efficiently. So a lot of problems in the computer science world can be solved, but they can't be solved in a quick manner. And so I'll get into what quick means in a second. But essentially the contests come down to trying to figure out an efficient solution to a problem, implementing the efficient solution to a problem, and then getting the problem right. So. And so as a brief relevant problem related to competitive programming, so we've actually had this problem in a competition before. So given a plane, a 2D plane with a bunch of coordinates in it, trying to find the triangle with the largest area that's formed from a set of 3 points. And so on the left side here, you can see that it's really not too difficult, like from our eyes, to see what the largest triangle is when you have like a small amount of points. But on the right side, for example, if you have a ton of points, A, it's pretty hard for us to figure out, and B, it's actually non-trivial to come up with an efficient solution for solving this problem. And so an inefficient solution would be to take every set of 3 points that you have in the graph and then finding its area and then checking to see if it's the largest area. And so that does solve the problem, but it's actually incredibly slow. And so there are faster ways to solve this problem, namely pretend that you wrapped a rubber band around the set of points. The rubber band would form to the shape of the points on the outside of all the other points. And actually, it turns out that in this case, the largest triangle, all 3 of its points will lie on this outside shape. And that algorithm for finding that outside shape, it's called the Convex Hall algorithm, and it's incredibly important for a lot of different reasons, and it has a ton of different applications in both the real world and in research, including the traveling salesman problem, computer graphics, and then like collision detection with automating cars to drive themselves. And so some benefits and like quick takeaways really quick. So within competitive programming, you need to have a really good understanding of different algorithms, different data structures, and then you also need to have a really good grasp on number theory and graph theory. So these kind of sound scary, but at the end of the day, because you're competing against other really good schools, you need to have this knowledge in order to succeed and do well. And so with that, one of the biggest reasons why competitive programming actually has benefits towards research and towards the real world actually is because a lot of the techniques that you learned are not commonly taught in undergraduate courses. And so actually some of the data structures that you need for and algorithms that you need to compete competitively in competitive programming are more so taught in our graduate level algorithms course here at UK. And so with that, having that knowledge before getting to that graduate level course, or just having that knowledge in general, is definitely beneficial towards your research at the end of the day.
Competitive programming also increases your skills in problem solving, because you'll need to be able to quickly analyze the problem, figure out what the problem actually is, and then figure out a way to solve that problem. And it also increases your skills in algorithm creation, as in each problem you'll have to figure out how to write that algorithm. And it also increases your efficiency and speed for coding, as the goal is to get these problems solved as quickly as possible. It also increases your critical thinking skills, because you need to have a deep knowledge of your data structures and algorithms and how they work, so that way you know how to apply them to the different problems that you'll be receiving. And it also, your algorithm and data structure creation, you need to not only know how those data structures and algorithms work, but you need to know how to create them in your code. And you also need to know if you actually solve the problem, because a lot of times you'll think that your method actually solves the problem, but then there's like something that you're missing, and so you get it wrong. So you need to make sure that you're actually solving the problem that is given to you.
Yeah. And some of the biggest reasons why those things that Chase just said apply to competitive programming and to research is because when you are in competitive programming, you get just a general knowledge of these data structures. And so having knowledge of different data structures or algorithms is definitely important when presented with a new problem within research, because then you can kind of look into your mind and try to figure out different ways that you could efficiently solve problems. And so with that, some other benefits to research include just like personal traits in general. So personally, for competitive programming, I probably spend about 10 to 20 hours a week on it. And so with that, you kind of get used to just having a big time commitment towards something, which is definitely important towards research or academia if that's your goal. And so getting used to that time commitment and getting used to spending vast amount of time thinking about problems and practicing how to solve problems is incredibly important. Additionally, you get experience working with high performance teams on managing tasks. So in these competitions, they're usually 3 to 5 hours long, and you have a lot of problems to solve. And so you need to figure out how to optimally manage your time so that not only you can solve all the problems, but that you can solve them in a good amount of time / faster than the other teams. And so with that, just like in the research world, trying to work with other people is something that you really get a good grasp on from competitive programming. And then again, just having like a general increase in your efficiency and speed of solving problems is always something that's great to have. And so some benefits of competitive programming, right? So more research related. So RU specifically. So I know on my team, we had a student that had an RU at the University of Southern California last summer, as well as another student who had one here at the University of Kentucky. And then that's among others, though. We have had other students go to other RU positions. And so with that, just being on the team helps you get those positions so that you can actually perform research in your free time. And then additionally, it also helps you for that next level of research at graduate level programs at Ivy League schools such as the University of Pennsylvania or Georgia Tech. And so with that, just having the differing knowledge of how algorithms and data structures work and then applying that to research can definitely help you get into these programs.
So there are lots of courses here at the University of Kentucky that help with competitive programming. The first one that you would probably take in your CS track would be CS 315, which is Algorithm Design and Analysis. And that class is a really good course for giving you your first introduction to algorithms and data structures. In that class, you'll learn a lot of like major algorithms that are very useful in competitive programming, and that also will come in handy when it comes to research as well.
Exactly. And then so 515 is the graduate level algorithms course. And so with that, that covers a lot of proofs that you need to understand for that. And in the field of research, especially with mathematics and computer science, there's a really big emphasis on proving mathematically why your solution is optimal or why it works. And so with that, that course and competitive programming both help build a foundation for that research aspect. And then also, so CS485G was a class taught by Dr. Yaramchek in the spring of 2020. And so with that, we covered different uncommon algorithms and efficient implementations of these algorithms, as well as proofs for why they work, and working on our personal development as to proving as to how those proofs work. And then so a lot of courses, though, are actually related to competitive programming. So basically, like any theoretical or applied computer science course or mathematical course really helps. So some examples include 415, which is our graph theory course, 416, which is our optimization course, or even CS or mathematics 340, which works a lot with number theory as well. And then, so at the end of the day, it might seem like competitive programming is more geared towards like computer science or computer engineering students, but it's actually really geared towards everybody. There's a lot of theory that comes from mathematics and other majors that you need for it. And so some of the other majors that we've had from other members, you can see them on the screen, but just to name a few, like physics, chemistry, mathematical economics, like those might not seem like majors that you would think that we would have, but we do have members with those majors. And so it is definitely has a very broad scope when it comes to who's on the team. And the reason why that broad scope is important is because a lot of these different algorithms actually apply to different areas of research. So for example, with economics, right, Hall's marriage theorem is really important for economics and like grouping different pairs. And with that, like learning about that and just having the general knowledge of that to apply to your own research is definitely important, as well as like computational biology. That's pretty computer science related, and it definitely has a bridge with biology, as you can see by the name. But yeah, so just in general, a lot of these majors and different areas of research all come back to computer science, and competitive programming really helps pad you, and it helps get you ready for this research. So I think that's the end of it. And at the end, we would like to thank Dr. Yaramchek for helping us and sponsoring us throughout the entirety of our season, as well as serving as a mentor for other students on the team that do research.
All right, thank you so much. There is a question here from Dr. Yaramchek. He says, this talk was about improving research abilities by solving non-trivial synthetic or abstract problems. Could you comment on how competitive programming plays or has played a role in your academic and real life work?
For sure. So I guess to answer the real life part of it, personally, I have accepted internship positions with Dropbox and Amazon for next summer and fall. And I think that doing competitive programming really gave me a good foundation for solving algorithmic puzzles, which they ask you in interviews. Additionally, I know other students as well on the team have gotten like RU positions. I know Chase applied for some in the fall. So with that, or not RU positions for the fall, but just like research positions in general. And so with that, it really just helps get your general knowledge on those different areas, and it really helps you get ready for interviews and things like that.
Chase, do you have any other, I was gonna say, you have a comment on that?
It also helps you in your other like CS classes, just because you're getting a deeper knowledge of how these algorithms and data structures work. So when you go up into the higher level courses, you'll have a better understanding, and it kind of gives you a step up when you're doing your work.
Okay. Are there any other questions? How did your team do this year?
So this year we got past the regional competition. So our region is like Vanderbilt, Chicago. And then so we got past that to the top. I forget how many teams qualified, but then so there's 2 rounds of like the national competition. So me and 2 other students competed at the first round of that, but one of them actually couldn't compete. So it was so we like had another person step in for them, but we weren't able to qualify to the next round. Shashank was actually Shashank, he was the person who like couldn't compete. He was our main like number theory guy for like math, and some of those questions had to do with number theory. So like as a team,
You need a
because
full team, all the skills on the
exactly,
people
yes, we are kind of lacking in that department. So we didn't get to the next round.
So one more question here is, what online training problem sets do you use, if any? USACO?
Yeah,
Question?
so Shashank does US ACO, and then I personally do Leak Code and Caddis and Code Forces. Code Forces has competitions, I think, every week to every other week. And so with that, it's just, they're basically the exact same. So like what we do here is collegiate level competitive programming. And then so those other ones that were mentioned include high schools, but it also includes like people that have already graduated college. And so you get to experience competing against other people. So there's always like, they give you room to improve essentially.
All right, well, thank you very much.
Thank you.
And now we'll move on to the last speaker in our session, which is underneath in the math category. So Philip Mearsman, you ready to go, Philip?
Yes, I'm ready.
Excellent.
Is my presentation visible?
Yes, but I'm not sure we can see all of it.
Is this better?
On my screen, it looks too big. I can't see it all. I don't know about other viewers.
It looks okay on mine, Martha.
Okay, so maybe it's I'm on my laptop. Maybe it's too small. Okay.
Well, if you have any questions about things you can't see, feel free to ask. So yes, I'm Philip Mearsman. I have been doing research in the math department on using semi-algebraic parametric analysis by metaprogramming, or SPAM, in portfolio optimization. I've been advised by Dr. Joe from the mathematics department. And hearing the last presentation, some competitive programming background probably would have been useful in my project. So I'll first give a brief overview of SPAM and what semi-algebraic parametric analysis is. Broadly, it's a programming project written in Sage Math, which itself is a mathematical software system that's based mostly on Python. And SPAM is a collection of algorithms and functions written in Python, which allow the solution to a very general set of computational problems in mathematics. And specifically, SPAM allows us to solve parametrized problems in a more comprehensive way than has been possible. And one of the important outputs of SPAM, or results of using SPAM to solve mathematical problems, is the generation of a proof cell complex, which provides a sort of computational proof for a parametrized problem in mathematics, as I mentioned. And in using SPAM, it's beneficial because it provides a faster and more informative solution, specifically to portfolio optimization and in other parametrized problems as well. So as an example of SPAM and how it works, consider a small 2x2 matrix with entries as displayed. And a natural question that we might ask is, for which values of A and B is this matrix positive definite? And we might posit that A is positive definite if and only if this inequality holds. And it's important to note that this inequality is easy to evaluate if you're given a value for A and B. You just plug in A, plug in B, and then see whether the left-hand side is greater than the right-hand side. However, we might desire a complete solution for every set of parameter tuples, A and B, that define this problem. And so SPAM helps accomplish this. And without going too in depth into how it works, this red and blue plot is an example of a proof cell complex that SPAM generates. So everything within the blue cell of this complex, every pair AB, which lies in the blue cell, generates a positive definite matrix. And everything in the red cell, if you choose a coordinate pair A and B that lie in the red cell, matrix A will not be positive definite. So this is a graphical representation of some results that SPAM generates for us. In portfolio optimization, which is what my project was focused on, SPAM is also useful. So portfolio optimization broadly is the allocation of funds to different portfolio assets, usually with the goal of maximizing one's expected return in mind. So you can think of this as, you have a selection of stocks that you want to invest in, and you're wondering how best to invest your money, depending on how risk averse you are or what your acceptable level of risk is. So a representation of what one of these problems looks like is displayed here. As you can tell in the objective function, or the first line of that problem, you see mu appear in the objective function. And mu here is the parameter representing the mean absolute deviation from mean, which is a measure of risk in portfolio optimization. And so again, similar to the positive definite matrix example, this problem is easy to solve computationally with a concrete value for mu given. However, we might want to solve this problem for every possible value for mu. And this is a more difficult task, and it's something that SPAM automates for us. So first, the traditional method for solving these problems with parameters is the parametric simplex method. And broadly, it's sort of the textbook method for solving a linear program with a parameter. And it can be applied to this portfolio optimization example. And it results in quote unquote dictionaries like you see here. And the parametric simplex method works. However, we're looking for a faster and automated method to solve these problems. And so we have used SPAM. This is an example of what some of our code looks like, just def statements. If you're interested, these link to our GitHub repositories where all of our code is stored. So this is sort of basic Python syntax. This is what it looks like when we're defining the functions that we use in our research, and an example of what some of our output looks like. So in the small portfolio optimization example I showed in the previous slides, these outputs are applicable to that problem. So in interpreting this, the first interval you see from 0 to 1.28 defines the range for mu for which the portfolio listed is optimal. So if you're deciding between these three different assets in terms of investing your money, and your risk measure, whatever that might mean, lies between 0 and 1.3, you'll want to put all of your portfolio into the third asset. And so in rediscovering some textbook results, we consider a larger linear program with 9 different assets available and historical data from 24 different time periods. And this is the output generated by SPAM, generated with our method. And as you can see, all of the different colors in this plot represent different assets that one could invest in. There are 9 different colors. And reading the graph from left to right, as you travel to the right, that means your acceptable level of risk is increasing, or it could correspond to somebody who is less risk averse than a mu value that's closer to 0. And so interpreting this plot, as you start from the left, you see bonds in red takes up a significant portion of one's portfolio. So this makes sense and is expected because bonds are generally considered a low risk investment. And so the low risk investor will put a lot into bonds. As we travel to the right and head towards mu equals 10, it's interesting to note that at about 8.2, mu equals 8.2, one's optimal portfolio would consist completely of stocks in the energy sector. These are sectors in this example. And so this does a nice job illustrating visually what this problem means. And then I took our SPAM methods and applied them to some real world data. So I used historical returns from these 9 well-known stocks and 1 5-year corporate bond, and ran the same method, used SPAM, and came up with these results. And so interestingly, in this plot, we only see 5 different colors, and 5 different colors despite there being 9 possible assets to invest in. And this is because 4 of the assets are dominated by the other 5. in this range of mu from 0 to 10. Also interesting to note is Bitcoin's massive amount of area on this graph. And so as we travel to the right, or as an investor becomes less risk averse, more risk loving, their optimal portfolio will soon consist entirely of Bitcoin. And this makes sense because we know that Bitcoin has a huge possible return. However, it's a pretty risky asset currently. And so, more traditional stocks like Nike, Microsoft, or Tesla lie on the less risky part of the graph and comprise most of a portfolio for somebody who is more risk averse. And so in the future, this SPAM could be applied to many more quantitative finance problems or problems more generally in economics, specifically problems that are parametrized, whether it's a risk aversion parameter or a savings rate parameter or anything. SPAM works on a very broad class of problems in economics. And so this is what I have for my presentation, and I'm happy to take questions.
All right, thank you. This is just theoretical though, right? So you're not going all in on Bitcoin right now, are you?
Oh, yes. I should note that I'm not a financial advisor, so invest at your own risk.
Yeah, you always have to have that as a disclaimer. Are there any questions here?
It looks like he's got 3 in the Q&A.
No, that was from the last talk, I think, or the ones I see are from the last presentation.
Oh, okay.
Unless you're seeing some new ones.
Nope, I thought they were new.
Okay.
Okay, so Philip, from your, I'll ask you a question. From your research, was there anything that was especially surprising to you?
I was surprised, particularly in this, in the replication of the textbook results, surprised at first by how quickly bonds and staple investments sort of trailed off as optimal investments in a portfolio. And obviously this is a small example, only considering 9 different investments, but it's interesting how quickly one's optimal portfolio might change for a small change in their level of acceptable risk. So everybody knows that investing is risky inherently, but it's interesting to see, how quickly your optimal portfolio changes if you become even a little bit less or a little bit more risk averse.
This makes me want to get a little riskier in my portfolio.
Yes, as Dr. Peterson said, I should make that disclaimer that I am no advisor.
So on your last slide where you selected what you had 10 or 8 or 10, I guess Apple doesn't even show up there, but did you select these for any particular reason, or you just took these and because you had some historical data on them that you could put into your algorithm?
I specifically chose these because they're well known and I thought they would be interesting in a presentation for people to identify stocks and assets that they're familiar with. And then I chose some of the most successful stocks as well. So we've seen Bitcoin and Tesla obviously do very well recently.
Right. I've done well with Apple, but I've had it for probably 30 years. So
yeah.
I was an early believer. So any other questions from the audience?
So I have one other question for you. Philip, what are your future goals?
I have accepted a PhD candidate or PhD student position, I suppose, at Vanderbilt University in economics. So I'll be starting this fall in their PhD program.
Very good. So pretty soon you really could say that you could offer advice.
Perhaps, yes.
Really good. So,
well, I just wanted to thank your mentor first and foremost for reaching out to make this presentation possible.
Yes, thanks for squeezing me in last minute.
Absolutely. And if Do any of our panelists have any last minute questions? Okay, I'm trying to switch gears. Okay, well, I want to thank you all for your presentations, for your research. I want to thank the mentors in attendance for their dedication to supporting undergraduate research initiatives here at UK. I especially want to thank Dr. Martha Peterson for being moderator extraordinaire for our final session of the 15th annual Showcase of Undergraduate Scholars. Thank you all for all of your hard work. Good luck with the rest of your semester. And this officially closes the showcase. Have a great
Wow, this is the last one, isn't it? Excellent.
It is. It
Excellent.
is.
And yeah, to the students, good luck with the rest of the semester. And it's winding down fast.
Thank you all.
All right.